the position of the nucleus. Generally it remains at the central part of your cell, and inside the nucleus you can see one bulging part is showing at here in that ultrastructure. Look at here, and that part is known as your look at here, that part is known as your nucleolus. Yes, very good, it is known as nucleolus, and it doesn't have any membrane around it. That means around the nucleolus there is no membrane. Generally the nucleolus contents gets connected with the nucleoplasm, that means the matrix of your nucleolus. Now you can see here that in case of some organism, you can see their cell consists only one cell, and in case of some cell there may be more than one cell, and in case of some cell which are completely devoid of in case of some cell it consists of only one nucleus, but in case of some cell which contains number of nuclei, okay, nuclei, the plural of nucleus is nuclei, remember. And look at here, this one is the RBC, this one is your RBC, and it is completely devoid of your nucleus. At the initial stage of RBC it contains nucleus, but at the final stage it remains completely devoid of nucleus. Okay, then look at here, you can see here that your nucleus is surrounded by two membrane. One is outer membrane and another one is the inner membrane. The outer membrane, in between these two membrane there is the intermembrane space, and which is filled up of some specialized fluid material, and that inner membrane and the outer membrane, they when joins together they forms the nuclear pore through which the nucleic material can pass through the outer part, that is your cytoplasm. Okay, through the nuclear pore you can the nucleoplasm releases its content to the outer cytoplasmic region. Okay, at that point only both the inner and outer membrane gets connected with each other. Then you have to proceed. You can see here that inside the nucleus you can see the dense structure is present, that is your nucleolus, and your nucleolus is consist of ribosomal RNA along with some ribosomes and some proteins. Okay, and as you know that RNA generally contains the code for amino acid arrangement, and when such amino acids get arranged by ribosomes they develop the proteins. Okay, and in case of the cell which have greater capacity to produce protein, they have number of nuclei. As you have already learned that the cell which are going to produce more energy, they have higher number of mitochondria. Similarly, the cell which has the capacity to produce more number of protein, they have the higher number of nuclei. Now look at here, your chromosome. That now return to your nucleoplasmic content of the cell, in which is present at the inner to the nuclear envelope, and that contains the chromatin fibers or your chromatin threads. Look at here, these chromatin threads, and these red color parts is your chromatin thread, and that thread, look at here, they are bounded around certain proteins. Okay, and that proteins are called as your histone proteins. Except histone proteins, some non-histone proteins are also there. Okay, and simultaneously they help to aggregate the chromatin fiber in such a manner that they can fit perfectly inside the nucleoplasm of your nucleus. You can see here that. And another thing you have to remember, during the course of cell division, that means when the cell undergoes cell division, At that time, your chromatin fiber condenses to form chromosome. Look at here. In this diagram, you can understood perfectly. Look at here. Here is the chromosome, and that chromosome contains your DNA thread or your chromatin thread. Okay, look at here. These are the chromatin threads, and if you will view such part, you can see. Look at here. You can see, if you view only this much part, you can see here that these are highly arranged. Look at here, they are highly arranged with the histone proteins. Look at here, these are histone proteins, and around the histone proteins the chromatin fibers are rounded in such a manner that they form the bead structure. Bead, you know. Then yes, these are the histone proteins. Then look at here. Then if you again unfold that chromatin fiber or your DNA fiber from the around the histone protein, you can see here that if you unfold the DNA chromatin fiber, you can see, look at here, how your DNA is looking. Look at here, this one is your DNA, and when DNA becomes thread like structure, it coils around the histone protein, and along with the histone protein some non-histone proteins are also there, and this non-histone protein, histone protein, and your chromatin fiber, they all, these are the histone proteins. Okay, they all aggregate in such a manner during the course of cell division by the process of your condensation, they forms your chromosome. Okay, this one is the structure of your chromosome. Take the screenshot. Now look at here. Here is the structure of your chromosome, and you can see that your chromosome has one centrally constricted region. This part is known as your, it is known as your centromere. Don't be confused with centrosome. It is the centromere. Okay, look at it. That constricted part, middle part, is known as your centromere, and around to the centromere two disc-shaped structures are there. They are known as kinetochore. Okay, look at here. Two disc-shaped structures are there. They are in generally green color. Okay, look at here. Here in this diagram, it is showing in the green color, and this is your kinetochore. Got that? And this one is your DNA. No, that is one notch around your chromosome. Okay, now look at here, and your chromosome now has two arms. Okay, these are the arms of chromosome. One is upper arm and one is the lower arm. Okay, now on the, we have to learn about, there are various types of chromosome which are found in the nucleoplasm of the nucleus. On the basis of the position of centromere in the cell, we can see that position of centromere, that means this part, we are going to classify the chromosomes into four main types. Look at here. These are metacentric, submetacentric, acrocentric, and telocentric. In case of metacentric chromosome, you can see that the centromere is exactly remains at the middle part of the two arms or the chromosome, which produces two equal length of arms of one chromosome. Okay, and in case of submetacentric chromosome, what happens? The centromere remains closer to any one arm. Look at here. So it forms two arms which are of different lengths or unequal lengths. In case of acrocentric chromosome, the centromere remains very closer to one end. Look at here. The centromere remains very closer to one end, so that there are two different types of arms will form from the chromosome. One is extremely short and one is very long comparatively to the shorter one. Then another type of chromosome, that one is the last chromosome. It is your telocentric chromosome. In case of telocentric chromosome, you can see that the centromere is located at the last end or at the one end of the single arm. So here you can see only one single arm is present in telocentric chromosome. That's all about your types of chromosome. Okay. Now sometimes what happen, except one constriction that you are seeing, this one is the primary constriction and you are calling it as the centromere. Sometimes the chromosome has secondary constriction which is present at any one end of the chromosome's arm or any single arm, and that constriction forms the one notch like structure that is called as your satellite, and that satellite generally involves in the formation of nucleolus. Okay, that satellite involves in the formation of nucleolus, which and that process occurs after cell division. Remember that satellite involves in the formation of nucleolus after cell division. Then we have to know about what is the function of your chromosome. Generally, chromosome contains genetic code inside the DNA and it helps in various activities of the nucleus. Now take the screenshot for your note purpose. Take the screenshot. If you have any doubt, now take the screenshot for the functions of chromosome. If you have any doubt regarding to this class, you can ask me later. Take the screenshot for the function of nucleus. Okay, very good, fine. So we have completed your chapter. Now we are going to discuss about some practical skills, okay, that related with your osmosis. And as you know that for your better understanding, I want to recall the method of osmosis. Osmosis is the movement of water molecule from its higher concentration to the lower concentration region through the semi-permeable membrane. Okay, now look at here. Generally, your solution are of three types. Remember, solution are of three types. The solution is generally made up of solute and solvent. Your solution is generally made up of solute and solvent. Solvent is the liquid content of the solution. Whereas, for instance, if you want to make the sugar solution, you have to add the sugar particles, which are the solutes, whereas the water content that you are taking, this is the solvent content. Okay, water is your solvent, whereas sugar is your solute. Now, on the basis of the concentration of solute in a given solution, you can categorize the solution into three different types. These are hypertonic solution, hypotonic solution, and isotonic solution. Hyper means higher. Remember, hyper means your higher. Okay, that means the solution in which there is higher concentration of solute than the solvent. At that time we can say that this solution is hypertonic solution. Look at here. Here the yellow color structure is your cell. Consider this one is your cell, and this cell is remaining inside a hypertonic solution. That means that container contains more amount of NaCl. NaCl stands for sodium chloride salt. Okay, more amount of sodium chloride molecules than that of water molecule. So that this type of solution is hypertonic solution. Look at here. In case of hypotonic solution, what happens? Here you can see only two sodium chloride molecules are present, which are comparatively less molecule than that of the water content, so that they are called as hypotonic solution. Hypo means below or low. Okay, here the solute concentration is very less compared to the solvent concentration. Solvent means your water. Now in case of isotonic solution, you can see the solute concentration is similar to the solvent concentration. Here here you can see the solute concentration is similar to the solvent concentration, so that this type of solution is the isotonic solution. And here you have to make compare with the cell that contains such type of solute or not. Okay, if you will put a cell, and as you know that during the course of osmosis, what happens, the water moves from its, look at here, as you know that the plasma membrane is selectively permeable in nature. Look at here, this one is the structure of your plasma membrane. The water can transport through the aquaporin protein, channel protein. This protein is called as aquaporin channel protein. The water molecule can transport from its higher concentration region to the lower concentration region through the aquaporin protein, which is present in the cell membrane. Okay, and when it will occur, when there is more amount of water is present outside to the cell comparable to the water content inside the cell. Okay, in case of, look at here, I would like to show the previous slide. In case of hypertonic solution, look at there, the solute concentration is more comparatively the solute concentration present inside the cell. Here look at here, only 2 solute molecule is present inside the cell, whereas there are nearly about 8 solute molecule is present outside the cell. So here this type of solution is hypertonic solution, and the water molecule is more inside the cell. So what will happen, the water molecule will come outside to the cell. Okay, so that what will happen, the cell will decrease its volume. In case of hypotonic solution, what will happen, look at there, the solute concentration is less comparatively to the solute concentration present inside the cell. Okay, and here you can see that the water molecule is more which are outside to the cell. Okay, and at that condition what happens, the water molecule will enter into the cell. Okay, so that what will happen, the volume of the cell will increase, because more amount of water will rush into the cell and it will increase the volume of the cell. And listen, in case of isotonic solution, what happens, look at there, the solute concentration of cytoplasm, that means inside the cell, look at here, is same as to the solute concentration present in the solution. So that in case of such type of solution, that means isotonic solution, what happens, the water cannot move from one place to another place, because there is no concentration gradient, concentration difference. Okay, water molecule remains same, and the solute concentration, that means sodium chloride concentration also remains same, so that there will be no movement of particles, water molecule from any zone of the solution, or you either from the solution or from the cell, so that the cell size will remain intact. Okay, and look at here, so this is the condition what will happen if we place a cell inside the hypotonic solution, and if we place the cell in hypotonic solution, and in case of the isotonic solution, what will happen, look at here, the cell generally shrinks in size when it remains inside the hypertonic solution. Hyper means more solute concentration. If the solute concentration is more in the solution, at that this type of solution is called as hypertonic solution. And if we place our cell into it, then our cell will lose water and it will decrease its volume, and that condition is called as plasmolysis of cell. Understood? And when the water molecule will rush into the cell, by keeping the cell inside the hypotonic solution, at that time what will happen, the water molecule is more outside to the cell, so that the water molecule rush into the cell along with the concentration gradient, so that the cell will increase its volume. That means it will swell up, so that condition of the cell, or that process through which the cell swells up because of the osmosis, is called as cytolysis. Sometimes what happens in case of animal cell, though the animal cell has no cell wall, so it doesn't bear the pressure of increasing volume, it can burst. Okay, after about 1 day or 2 day, or nearly about 8 to 10 hours, the cell will burst. Okay, but in case of plant cell, it has one additional covering, that is your cell wall, which maintains that pressure, but the cytoplasmic content remains swelling up. Okay, in case of, now come towards the last type of our solution, that is in isotonic solution. What will happen? The cell will remain its volume unchanged, it keeps its volume same, because there are really what equal amount of solute and solvent concentration compared. If you will compare inside the solution of your cytoplasm as well as the outsider environment of your outsider concentration of your solute and solvent in outsider solution. Okay, but here what will happen practically, if you will place red blood cell, which is one animal cell, and one plant cell, which is have the cell wall around it, what will happen in case of isotonic solution? Look here, the reverse has given. In case of isotonic solution, they both will remain its normal size or normal volume, because the cytoplasmic concentration remains similar to the outer solution's concentration. In case of hypotonic solution, what will happen? The animal cell will swell up, and even it can burst by keeping it for more time. Then in case of plant, what will happen? The cytoplasmic content of the plant cell will swell up, and it will create pressure upon the cell wall. And one thing I want to say here, that the pressure that develops inside the cell is caused by osmosis, is called as osmotic pressure. It is called as osmotic pressure. Whereas when that swelling content of the cell creates pressure upon the cell wall, that is called as osmotic pressure. Remember, at the same time, the cell maintains its strength or turgidity because of its hard cell wall. As you know that plant cell wall is made up of cellulose and hemicellulose and other components, so it will maintain its strength because of its turgidity, and the cell wall will also against create pressure to maintain its strength, okay, against the growing pressure because of osmosis. So that pressure against the cell wall, that pressure created by cell wall upon the cell's cytoplasmic content is called as Turgor pressure. Okay, remember that pressure is called as Turgor pressure, Turcer, Turgor pressure. And Turgor pressure is created by cell wall to maintain turgidity, so the normal cell becomes turgid. Okay, remember, the normal cell becomes turgid and normal plant cell. But in case of animal cell, it doesn't have any protecting cover around it, that means no cell wall, so it will burst off. In case of hypotonic solution, what will happen? The cell will lose its water. Okay, that means there will be net loss of water and the cell will shrink its size. Okay, and that type of cell is called as shriveled cell. But in case of plant cell, what will happen? That in case of plant cell, the plant cell also shrinks its size because of the loss of water to the hypertonic solution which has more solid concentration in it. That means in the solution, rather comparable to the solute present inside the cell. If there will be more solute in the solution, then solution will need more amount of solvent to make equilibrium, okay, to bring equilibrium. So that's all about your today's class, and have a nice day. Thank you. If you have any doubt, then you can ask. Okay.